All right. It's Thursday noon, and we are actually in week 8 of Bio 1140 here at UNM. And this week is test 2. And test 2 covers chapters 4, 5, and 6. So chapter 4 is the macromolecules. Chapter 5 is on cell biology, just like the general structure of cells. And chapter 6 is on energy and membranes and transport and chemical reactions. So for a lot of people, this test 2 is one of the harder tests of the semester. And in fact, for, oh my gosh, like over 10 years, the class average for test 2 has always been the lowest of all 4 tests that I give. And fortunately, test 3 and 4, the average is much higher. In fact, test 4, the highest is the highest average. Okay, so this is your chance here. This test is pretty awesome. There's no time limits on it. So you can open it up. You can go research your 20-point essay question. You are more than welcome to have the OLA for your section. Help you with some suggestions and comments before you submit that your essay to your test and submit your test. So I've had a couple of people about the material, and there's this general theme of people not knowing stuff. And I just got a question, and well, you know the essay question beforehand. Yeah, I sent it an email exactly how to answer it. It's the essay question is, what is the most interesting or important thing you learned in the last 3 chapters? So that's chapters 4, 5, and 6, and explain why. And then the second main part of that essay is to go add additional knowledge. And there's an email, and when you open up the test, you will also see the essay question with the question and the breakdown of the grading. And the big thing here is that 20-point essay question, 5 points is going to be grammar and spelling and clarity. How well did you write it? 10 points will be the extra stuff you looked up for that essay question. So I'm putting a much larger emphasis on looking things up, adding information not in the required reading. And then 5 points of that is, what is the most important or interesting thing you learned? That being said, let me get back to, thanks for the question, by the way. So if anybody else has another question, feel free to shoot it in here with the chat. So good question, Jacqueline. Okay. One of the issues, I'll go ahead and answer your question too. Awesome. Why are fatty acids hydrophobic? And this is a good question, actually, Savannah. That's a really good question. It's going to go into what I was about to say, so why this stuff is hard. And one of the reasons why is going from memorization to understanding. And that question of why fatty acids are hydrophobic is a great understanding level question. You can memorize they're hydrophobic, but what does that mean? So a fatty acid, let me see, I don't actually have a piece of paper near me. Okay, I'm sorry, I'm out of my element here. I don't actually have my chalkboard here or my whiteboard to write on, but imagine a fatty acid. Okay, it's a chain of carbons. So you got carbon, 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 and they're surrounded by hydrogens, right? So these carbons have hydrogen attached to them. So imagine a chain of carbons surrounded by hydrogen. Now, hydrophobic means it's water fearing. It means it cannot form a hydrogen bond with water. Okay, that's hydrophobic. Hydro means water, phobic means a fear of. So I've got all this carbon and hydrogen, and the chemical bond that holds the hydrogens to the carbon and the carbons to the carbons are covalent bonds. And specifically, those covalent bonds are what we say are non-polar. They're sharing those electrons equally. And because they're sharing them equally, this fatty acid cannot form any type of polarity. There's no way for it to form a hydrogen bond with water, so it moves away from water. It's hydrophobic because it just can't mix with it. And the reason why is because the carbon and hydrogen forms non-polar covalent bonds. They're sharing those electrons equally. Now you take something that's polar, like oxygen bonded to carbon, or nitrogen and hydrogen. Well, oxygen is way more electronegative, right? So it shares those electrons unequally, meaning the electrons spend more time near the oxygen. Okay, electrons are negatively charged. So if they're spending more time near the oxygen, it means the oxygen picks up a slight negative charge, And if it's bonded to hydrogen, that hydrogen or carbon or nitrogen picks up a slight positive charge. And we know that opposite charges attract. Whereas the fatty acid, there's no partial charges. There's, like I said, no way for these things to attract itself to water. So, Savannah, I hope I answered your question there. And that's a good one because that is the difference between just memorizing fats are hydrophobic, fats don't dissolve in water, and understanding why they don't dissolve in water. So good question on that one. And this goes back to, as I've talked to several people throughout the semester, or especially in the last couple of weeks, about not doing so in the class, there's this common theme that there's a lot of information and people aren't memorizing it very well. They're not committing it to memory. And when you don't have to study for a test, we kind of neglect our studies. But here's some advice I have for you on this test. Even though it's open book, open note, study. And you can study differently. But what I would do is familiarize yourself with the key terms. What are the key words? What are the key lecture or lessons? We can start with like chapter 4, the macromolecules. Okay, what is a macromolecule? Well, macro means big. These are large molecules. And there are 4 of them that you had to learn. And right now, you should be thinking, okay, carbohydrates, proteins, nucleic acids, and lipids. And then memorize that. That's not hard to memorize those. And then just know, well, what are their main purposes? Well, carbohydrates, obviously, for a quick source of energy. But in plants, carbs are also used for structure, right? They're used as cellulose to like make the cell walls really tough. Carbohydrates are also, as you break them down, they form precursors to other molecules. They're also used in our bodies for our cells to recognize our cells from other cells. So carbohydrates have a lot of different use. Okay. And then I'm going to come back, Justine. I just got your message. I'll come right back to that in a second. Proteins. Proteins are made up of amino acids. There's millions of proteins, and they form things like collagen. They form the cytoskeleton. Proteins have lots of uses. Lipids, which includes fats, waxes, oils, sterols like cholesterol, and your steroids, testosterone, estrogen. They have different purposes. So as you study, start with a really big picture, right? And as you learn the big picture, then start filling in the details. And like I said, I know because you don't have to study for the test and come into a class and take it. You might not, but that is one way to help you organize all of this information. Okay, so let's go here. How can we identify when something is oxidized or reduced? Okay, so we're talking about redox reactions. This is the end of chapter 6. And just real briefly, a chemical reaction makes and breaks chemical bonds. Okay, so I'm rearranging elements in the reactants, which is what I start with, and I'm going to break those bonds of the reactants, like let's say carbon dioxide or glucose molecule and oxygen, and I'm going to rearrange those into carbon dioxide and water. That would be a chemical reaction. Not changing elements, changing the molecules by rearranging the bonds between the elements. Now let's go on to a redox reaction, reduction oxidation. This has to do with the position of the electrons. Who's gaining electrons? Who's losing electrons? So if you're gaining electrons, you're being reduced. If you're losing electrons, you're being oxidized. So imagine you've got a very simple molecule like methane. Natural gas. If you guys burn natural gas, you know methane, ethane, propane. Methane is 1 carbon surrounded by 4 hydrogens. Ethane is just 2 carbons surrounded by 6 hydrogens. Propane is 3 carbons surrounded by 8 hydrogens. You get the idea. Okay, these molecules are sharing the electrons equally between the carbons and between the hydrogens. Okay, nobody's hogging the electrons. Now, when we burn gas, we break those bonds and we react it with oxygen in the atmosphere. And we form carbon dioxide and we form water. Okay. Oxygen is more electronegative than carbon or hydrogen. And the reason why it's more electronegative is because it has a greater pull on the electrons, right? Carbon's got six protons in the nucleus. Oxygen has 8. And as a result of having those 8, it's got a much stronger pull on the electrons. So when we take methane and we break it down to carbon dioxide and water, the oxygen is going to be reduced because it's going to gain electrons from the hydrogen to form water, or it's going to gain electrons from the carbon to form carbon dioxide because it's forming polar covalent bonds. Meanwhile, carbon and hydrogen are being oxidized because they are losing electrons. The electrons are moving away from them. And then, okay, here it is. How can I explain why oxidation and reduction always occur together? And it has to go with what I just said. We got, I think my kid's back there not having a good time. Luckily, my wife's helping out here. Okay, so if something's gaining electrons, being reduced, in this case, oxygen is being reduced, okay, then something has to be losing electrons, so something is being oxidized. So if something's gaining, something has to be losing. So if there's always a reduction, somebody's getting oxidized. And we can talk about cellular respiration. Glucose, or a fire, basically, you're taking glucose molecules, C6H12O6, in the presence of oxygen, and we're going to react that, and we're going to form carbon dioxide and water. Because glucose molecules have a lot of carbon bonded to carbon, carbon bonded to hydrogen, those molecules would be reduced. And then we react them with oxygen. Oxygen is going to gain the electrons to form carbon dioxide or water. And in that case, it's reduced. And as the hydrogens and carbons lose electrons, they are being oxidized. Okay, did I answer your questions? Let me know. And I'll keep going on here. Okay, I haven't seen back, so hopefully I answered your question sufficiently. And like I said, if anybody's out there and needs to ask a question, go for it. In chapter, let me go back here. Oh, thanks, Justine. Yep. All right, awesome. Good questions, ladies. Good questions. Okay, let's go on here. Chapter 5. Oh, you guys are asking questions. Awesome. Can I explain the concentration gradients have potential energy, and why does moving down the gradient increase entropy? That is a fantastic question. And that's right out of chapter 6, where I took all of these different things like energy and membranes and transport and weaved them all together into one story. Okay, so first of all, a concentration gradient. I need to really see if I can find me. I got to start out my paper today. Aha. I'll just use welcome to animation voiceover. Okay. Imagine a gradient, and I'm just going to write here 0 parts per thousand, 0 feet, and then up here we can have 5, 10, 15 feet, and 15 parts per thousand. Okay, so hopefully you guys can see this. I drew like a line right here, and here I've got 0 feet. So this is the bottom, and you can imagine going up a hill or down a hill. Here it's 15 feet high. And then here I've got a concentration gradient of 0 parts per thousand versus 15 parts per thousand. Okay, so if you're not familiar with parts per thousand, imagine I add in like 15 parts per thousands of salt to water. So we've got a concentration of 15 parts per thousand. What that means is if I reach in and grab 1,000 parts, I'm going to have 15 parts of it are going to be my salt, and the other 985 parts would be water, okay? Now, a gradient is I'm going to change a value over a distance. It's an elevational gradient. You walk up or down a hill, you're going up an elevational gradient because the elevation changes over a distance. Concentration gradients, the concentrations of my solutes. I'm adding salt to water, right? I'm adding a solute into my water. This has got lots of solvents in here. It's diet soda. Keeping me well every day, right? Every day I drink a diet soda. I don't get sick. Must work, right? Anecdotal data. Don't believe it. Okay, so here's my concentration gradient. Now, here's some of the relationships between the gradients and energy and entropy. Okay, so now we know energy. We can have kinetic energy, which is the energy of motion, and we can have potential energy, which is energy stored, and entropy is a measure of disorder. So if something's in equilibrium, if it's in a balanced state, you're high entropy. If you are in a state out of equilibrium, you have lower entropy because you have more order. And these things in lower entropy often have a lot of potential energy. So let's say I put a ball. I'm going to draw my ball right here. It's at the top of a hill. Sorry, this is super sophisticated here. This ball's at the top of a hill. It has potential energy based on its position of being at the top of the hill. Okay. If I let it go, the ball will roll down the hill and the potential energy will be transformed to kinetic energy. And when it reaches the bottom, the energy is all dissipated as basically heat and whatever. So now that it's gone from here to here, first law of thermodynamics, I still have the same amount of energy in the universe, but because it's now in equilibrium, that energy is no longer usable. We've increased entropy. Now let's talk about concentration gradients. This is very important for the test. I have a cell, and inside my cell is 15 parts per thousand, and outside the cell is 35 parts per thousand. Okay, because I've got 15 parts per thousand here, 35 parts per thousand here. There's a couple different things happening. First of all, we're out of equilibrium with our environment. That's good. That's where life wants to be, out of equilibrium. Okay, and then what happens is now we've got potential energy stored across this membrane. And in fact, our cells work very hard to move sodium ions out. You can see I've moved my sodium ions out, my potassium ions in, and what that does is create a membrane potential. And this membrane potential, right, I store potential energy here that we can use to do work, because I've got an electrochemical gradient. I've got 3 sodiums for every 2 potassiums. So that's a chemical gradient because these are charged ions. I get the outside of my cell is positively charged. The inside is negatively charged. So I have an electrochemical gradient. The system's out of equilibrium with its environment, and it has a low entropy. And because it's got this electrochemical gradient, it's storing potential energy. And then as things move across it, potential energy is converted to kinetic energy that cells can use to do work. Okay, so if I have a sodium ion flooding into here, it's moving down its concentration gradient. Okay, all right. And then just really quickly for the test, the other thing you'll want to know, if I've got 15 parts per thousand here, 35 parts per thousand here, this is hypertonic. That's Hyper, hypertonic. Water will exit the cell and it will shrink. And that's very important because the way that cells maintain water balance is by controlling their electrolytes. If a cell is losing water, it might move electrolytes in. And if the water is coming into the cell too much, it might move electrolytes out. So like I said, cells control their water balance by moving electrolytes. Okay, Sydney, you asked a really good question. Why can't we digest cellulose? I know cellulose is identical in almost every way to glucose molecules. And I'll just quickly draw a glucose molecule right here. But the reason why, the simple answer is we do not have the enzymes available. We don't have an enzyme to break down cellulose. And this to me is one of the mysteries of life, not being a hardcore biochemist. But this is a glucose molecule right here. Okay, that's glucose. And the difference between the one that makes cellulose and starch in a potato is the way I originally drew it. It's that little hydroxyl, that little OH, not much of an H. This molecule with this OH in the down position, that molecule, you stick them all together, you get starch like in a potato and or a carrot. However, if that little OH group is in the up position, this molecule will then come together to form cellulose. And to break down starch, we have specific enzymes that can break down the shape of that molecule. For whatever reason, animals don't make the enzyme to break down cellulose. And in fact, for some reason, having an enzyme to break down cellulose is incredibly rare. And it's been thought that it took almost 100 million years for life, bacteria, to figure out a way to break down cellulose in plants. And animals have never acquired it. I know that ruminants like cows, They say, oh, they can break down the cellulose. They actually cannot. Them and termites both have symbionts that live inside their gut that can break down cellulose. So it has to do with a small difference in the shape of the glucose molecule that is put together to form cellulose and our lack of an enzyme to break it down. Good question. Like I said, I need to go talk to some biochemists and understand why enzymes to break down cellulose are so rare, because cellulose is the most abundant organic molecule on the planet. Okay, any other questions here? All right. Now let me just continue on with chapter 6 really quick. If you've got a question, just shoot. Here it is. Chapter 5, before I get started, chapter 6. Chapter 5 is on cell biology. Make sure that you know the difference between prokaryotic cells and eukaryotic cells, and know something about the diversity of prokaryotes. One of the biggest mistakes people make every single time is plants are prokaryotes, animals are eukaryotes. That is not true. Plants and animals are both eukaryotic cells. We're defined as having a cell nucleus, but evolutionarily speaking, it's a mitochondria. And eukaryotic cells are larger, more complex cells that have the endomembrane system that are to help package proteins to be exported outside the cell, and also to help detoxify and break down things. So you want to familiarize yourself with those things as well. And eukaryotic cells also have a cytoskeleton. Cyto means like cell, skeleton. And that cytoskeleton has 3 components: microtubules, which are the largest. They're like hollow tubes. You can push a microtubule. Actin filaments, which are the smallest. Actin filaments are used as semi-permanent structures. Well, not semi. They're structures in the cell that can be broken down and rebuilt, just like microtubules. And importantly, they make our muscles. Your muscles are actin and myosin. And then the intermediate one is the keratin fibers, which are a group of proteins that are very strong and permanent inside of cells. And you're familiar with keratin. They form hair. Oh, I seem to be missing some hair here. And our fingernails. Okay, so make sure you familiarize yourself with the 3 parts of the cytoskeleton. Now, chapter 6. This one is hard for a lot of people because there's just a lot of information in there. But make sure you study the nature of energy, why life needs energy, the role of membranes in making energy, because that's important. Like, membranes are the gatekeeper to our cells, right? They control what comes in and comes out. So you want to know diffusion, facilitated transport, active transport. What's the difference between those? And then the importance of chemical reactions. And the main thing is start with the chemical reaction. I just explained it. It's making chemical bonds. And an important point, it requires energy to break a bond, and energy is released when chemical bonds are formed. And then we have these, and yeah, that's it. And then there's redox reactions I just talked about, which is the position of the electrons. Okay, so if anybody doesn't have any other questions, I'm going to go ahead and sign off. All right. Hey, for those of you listening, remember, you can take your essay question, you can send it to the OLA for the class, the CABS, and get feedback on your essay question, and then submit it so you can have it edited. And in all honesty, if you send it to each other and had each other edit it through the class, that's fine. I don't care. Have people look at it. Have people edit it. It doesn't matter. Just don't cut and paste it from somewhere else. That's all I ask. Just don't copy somebody else's work. But you're welcome to have other people look at it and in the class as well. And I'll make sure that the group conversation is on so you guys can talk about your essays if you wanted to. Is it open book? Oh, yeah, come on. Hey, Isabella. Yeah, it's totally open book, open note. Anything goes. Seriously. You can't cheat on this. No, seriously. I mean, if you just had somebody read off the answers to you, I'm not going to try to catch you. I wouldn't do that because I assume at some level you want to learn the material. I assume you're interested in some of it and you want to learn it. So open book, open note, and Google next to you and go look those answers up. That's fine. The test is not timed. There is no time limit. Take your time on it. Come open it. Come back to it. Enjoy looking up the answers. I'm trying to reduce the stress of these tests. I time the quizzes so you force you to learn a little bit, memorize a few things. But the larger 100-point open book test, even though it's more points, I really want you, I am encouraging you to go look the answers up and spend some time with the test and do well on it. And the other thing I want to say to you, to all of you with that test, like seriously, if you do not understand a question, write it down, screen copy it, I don't care. Send it to me with what you think the answer is and your reasoning for that answer, and we can talk about it from there. I have no problem discussing these test questions with you, but all I ask is you give me the answer and why you think that is the right answer. And like I said, we can open up a discussion from there. And so don't sit there and fret about test questions for hours if you don't understand. It makes sense to me as I wrote them, might not make sense to you. So like I said, I'm here for you to help out. I'll try to be around a little bit on Sunday, but other than that, I won't be around too much on Saturday at all. And I'll probably sign off around noon. Well, I won't. Okay, I'll spill the beans. My wife is 9 months pregnant and we have to go to a doctor's appointment tomorrow. So I won't be around too much on tomorrow. I'll be around first thing in the morning. But I'll try to make myself available Sunday afternoon for you guys, or Sunday evening. Yeah, 2 weeks, October 23rd. Our second girl's coming. It's crazy. Okay. Any other questions? All right. Oh, thanks. Yeah, I know. It's kind of exciting times. I'll announce to the class when she comes. You guys kind of got a spoiler. I don't think I've really told many people. Oh, cool. Carolina, can I explain channel proteins? Thanks, guys. I really appreciate the well wishes. Hey, so let's get back to this channel proteins. I'm glad somebody else asked a question. I enjoy answering your questions. This is kind of fun for me. Oh, and if anybody wants to do a Zoom meeting with me, let me know. I'm available on Zoom almost any day of the week to meet with you guys. Okay. Channel proteins. Okay, so let's talk about membranes and diffusion. So the membrane surrounds a cell and it regulates what comes in and goes out. So membranes have this hydrophobic interior. Now, what that means is membranes have selective permeability. Now, permeability, if something's permeable, it means things can flow through it. So a permeable barrier means water or oxygen or carbon dioxide can go through it. And that's exactly what goes through our membranes. But selective permeability means only certain things can go through it. So a channel protein is going to help things move through the membrane. So with our cellular membranes, electrolytes cannot pass through the membrane very easily because they are charged particles. The interior of the membrane is hydrophobic. So what we have are channel proteins. They form a channel, like a tunnel that goes through the membrane, allowing calcium, potassium, sodium, and chloride to move in and out of the cell through these channels. And that would be facilitated diffusion. Diffusion is spontaneous. It means you do not have to add energy. It just uses the kinetic energy that's already there. That's a good test question. Diffusion requires energy. You just don't have to add energy. So if we have facilitated diffusion, the channel protein, these are proteins that go, if you had a cell membrane, like this is our cell again. Sorry, I'll get my whiteboard back. If I have a membrane protein allowing sodium to go through here, that would be a channel protein. And like I said, they're involved with what is called facilitated diffusion. They're helping the diffusion of electrolytes into or out of the cell. And they're not just always tunnels. A lot of them they have gates on them. They can open and close those channel proteins. So those are called gated ion channels. Gated because they can open and close. Ions because they allow ions to pass through. And some are what we call, voltage gated ion channels. Voltage just means you're changing that potential across a membrane, which causes them to open or close. And some are called ligand gated. A ligand is just a chemical signal. So they open in response to a chemical signal. And so that's all there is. Okay. Any other questions here? And thanks for all the well wishes, everybody. I appreciate it. I'll post some pictures and keep you guys posted when I get back. But I'll be around for the next 2 weeks. But like I said, 2 weeks from tomorrow. All right. Anybody else? All right. Well, you guys have a great weekend. Like I said, I'll be available first thing in the morning, and I'll be available Sunday night. If you missed the Sunday night deadline, don't sweat it too much. I'll definitely be around all day Monday to discuss anything with the test that you need to. And you can turn the test in late and still get full credit. Don't worry about it. And I'm giving most people back at least 10 points in bonus points for the first half of the semester. Okay. Well, you guys have a great weekend too. And like I said, spend some time on that test and enjoy the being able to learn. All right. Well, you guys have a great weekend. You're welcome. My pleasure.